We are happy to have you. Floor is yours.
Wonderful, thank you. Can everyone hear me? Do I see any nods, hopefully? Great, perfect, thank you so much for that introduction. Thank you to all the organizers for inviting me today. It's a real honor to present my work to all of you. So my title here is a play on words, in that I'm gonna touch on different types of matrices throughout this talk, both physical and mathematical. But visually, this is a reference to the 1999 movie directed by the Wachowskis, which was my favorite movie as a teenager. And I think it's a good time to revisit it because it's basically about a hero trying to understand the rules governing the world around him so he can exert some control over it. And I imagine as scientists, we can all relate to that goal, even if it means something different to each of us. And for me, the underlying structure that I've been investigating for many years now are the gene regulatory networks controlling animal development. So I'm going to start with one of my favorite networks, which controls embryonic development in the fruit fly. So here you can see a baby fly that's about 2 hours old. And at this point, all of its cells look the same. But as gastrulation begins, suddenly these cells start doing really different things. So some are moving and folding up, others are growing and shrinking. And as time goes on, you'll see them start to form segments that make up the fly body, along with all the different types of cells that are necessary for it to live a happy life. And what I find really amazing is that all of this action and movement was actually set up at this stage here, where all the cells look the same, but they're not the same. They're already expressing different genes in very distinct patterns. And these patterns instruct each cell as to how it should behave moving forward. Now, this image is a visual rendering of quantitative gene expression data at cellular resolution. So this is the type of data that I collected as a graduate student in the DePace lab. And even though it looks like a cartoon, it's actually a really big matrix where each row is a cell and the columns list the expression measurements for different genes. And this matrix is incredibly powerful because it makes the embryo computable. So we can fit quantitative models describing how these patterns are built, and then we can test those models to learn more about the regulatory sequences that are controlling them. And that was the main focus of my graduate work. But for now, the embryo really illustrates how I think about gene regulatory networks operating in development. So gene expression patterns are carved out by regulatory proteins, often transcription factors, that act together to regulate their targets. And these targets can affect cell behavior by changing their shape or by inducing movement. I call these types of targets cellular effectors, and they include things like cytoskeletal proteins or adhesion molecules. And so in order to understand development, we need to figure out how these genes are connected within these networks. But these regulatory connections aren't only important for development, they're also important in animal evolution. So this is a classic example, also from flies, where this species in the middle has acquired this pigmented spot on its wing. And that same species has gained expression for a gene that deposits pigment during development. And this expression difference can be traced to changes within the regulatory sequences for that gene. And so essentially this new morphology evolved because a gene regulatory network got rewired. So some connections got stronger and others got weaker. And while this is one example, there are many case studies in the literature that show that mapping these gene regulatory networks can be a really useful tool for studying evolution. But what I've realized in my postdoc is that the opposite relationship is also true. So natural variation in animals can be a really interesting tool for mapping these networks themselves. Now, the actual networks that operate in development contain hundreds of genes and thousands of interactions. And so pinpointing the important ones for a particular process can take years of work. And so as a postdoc, I wanted to build an experimental system where I can study networks in the context of a trait that changes really rapidly between species. And because I spent so much time studying the embryo, if I could track those changes at the level of individual cells, then so much the better. And that's how I came to study fruit fly genital structures in the Rebbe's lab at the University of Pittsburgh. So here I'm showing the incredible diversity in male genital structures between 3 species in the melanogaster clade. I'm going to focus on a structure called the posterior lobe, which is outlined here in green. It's very different in all of these species, and I want to know why, especially between these two, Drosophila mauritiana and Drosophila simulans. So these species provide a really nice case study because they're really closely related. And even though their genitalia have diverged in shape, they can actually interbreed and generate fertile hybrids, which enables us to find genomic regions that are associated with differences in the structure. which are called quantitative trait loci, or QTL. And this is really important because this is how I'm planning to use natural variation in order to study regulatory networks. So these QTL can pinpoint regions of the genome that may have really interesting regulatory connections. But it's going to take me a few minutes to get there, and so I'm going to come back to this idea at the end of the talk. For now, I'm going to give these structures nicknames to save me time and to help you remember them. So the Mauritiana lobe is thin and spiky, so I'll call it the sword, and the sibilance lobe is big and flat, so I'll call it the shield. And basically, I want to find the genes that make the sword different from the shield. So to do that, I started by tracing their development. So these structures form from an epithelial sheet when the flies are pupa. So this is what they look like at a late stage, 48 hours after they become pupa. This is a sample from Drosophila murciana, where apical cell junctions are labeled in gray. And as I rotate the sample, you can see the sword here. But if I image the same tissue 20 hours earlier, this sheet of cells look much simpler because the sword and many of these other structures haven't formed yet. And so over this 20-hour period, this tissue gets warped into this really complex shape. So it has creases and folds at precise locations, and that's how it forms these genital structures. So let's compare this with Drosophila simulans. So as I rotate the simulant sample, you can see the shield projecting from the surrounding tissue. It's obviously much bigger than the sword. And at the same early time point, the shield hasn't yet started to form. Now, one can think of many cellular processes that could be building this structure. Maybe it's due to cell proliferation or cell migration, but based on some data that I don't have time to show, our lab figured out that the lobe is actually formed by extreme cell elongation. So basically, posterior lobe cells are just getting stretched. They're getting really tall and really thin. And more cells are told to do that in Drosophila simulans, and that accounts for the larger size of the shield. So now I could be a little bit more specific about my goal. I want to find the genes that are stretching lobe cells and then figure out why they're working differently between these species. So when I started this project, I assumed that the cellular effectors that are responsible for stretching cells are actually present inside those cells. So basically, I was thinking about cytoskeletal molecules that are jutting up against the apical membrane. Seems like a reasonable assumption. But midway through the project, we got some clues that the real culprits were actually outside of the cells as part of the extracellular matrix, or ECM. Now, while cell biologists think about this matrix a lot and know that it can exert force on cells in some cases, it hasn't really been considered a major player in the evolution of animal body parts. But of course, no one can be told what the matrix is. You have to see it for yourself. And here's what it looks like during the genital development in a different species, which is Drosophila melanogaster. So this data was collected from Sarah Smith, a former graduate student, and she was able to image a particular tagged matrix protein called dumpy, which you can see here in green. And she found that it forms this really elaborate surface that covers a bunch of genital structures as they're developing. And here you can see labeled dumpy right on top of the posterior lobe. Sarah also found that when you alter dumpy expression with RNAi, you change the shape of a lobe. And so these experiments together showed us that the matrix in general and dumpy specifically seemed really important for lobe development in melanogaster. And because of that, I decided to see just how deep this rabbit hole goes and test its role in lobe evolution using Mauritiana and simulants. And so I use fluorescent lectins to visualize the matrix during development because we don't have these nice tagged versions of dumpy in these species. So at this point, genital structures are in the middle of their development, and you can see that the matrix is associated with the lobe in both species, but I'm going to rotate and slice these samples so you can see that more clearly. So first, I'm going to slice at the lower boundary of the sword and then rotate it and gradually move this slice upward. And as I do that, you're going to see that the connection to the sword eventually goes away right about here. That's because there's no matrix present over there at that time. And as I rotate the sample again to see where that slice falls, you'll see that it's right at the upper boundary of the sword. Now I'm going to do the same thing with the shield. I'm going to rotate it again, slice at its lower boundary, and then start moving up. I have a longer way to go because it's a much bigger lobe, but eventually, again, you're going to see these connections start to disappear as the presence of the matrix goes away as you move upward, and it disappears nearly completely right here. And again, that slice is right at the upper edge of the shield. So from these movies, we can see that the matrix is associated with both lobes as they're forming and seems to make more extensive connections to the developing shield. But in order to see how these connections with the matrix arise, I needed to look earlier in lobe development. So in these samples, the lobe hasn't yet started to form. So I'm first going to rotate the Mauritiana sample to show you the surface. Then I'm going to light up the matrix in green here. And then when I take away the cell outlines, you can see that the ECM casts this net toward the cells that are eventually going to form the sword, and those connections only extend about partway up the tissue. Now, the situation is a little different in simulants. Here you can see the matrix extends further up the tissue compared to Mauritiana. And once I take away the cell outlines, you'll see that it lies right along the edge of the developing shield. And so in both species, we see this correlation between the size of the lobe and its association with the matrix, even before the lobe has started to form. And that suggests that the matrix plays some role in its diversification. So at this point, it seems like the matrix as a whole is associated with the developing lobe and looks pretty different in these species. But I wanted to know if I can pin any of these differences to changes in the gene expression of specific matrix components. And the gene that I started with is dumpy because it seems so important for lobe development. So here's the lectin data that I just showed you. And now I'm going to light up dumpy RNA in Mauritiana, which I labeled using hybridization chain reaction. And we can see that these patterns are actually really similar. So dumpy expression extends only partway up the tissue and seems to mark cells that are destined to elongate and form the sword. And when we take a look at simulants, again, this pattern looks really similar to the lectin stain. So the dumpy expression pattern has expanded, which may explain why the shield is bigger than the sword. So in my last few minutes, I want to come back to my general goal, which was using natural variation to map gene regulatory networks. Now, these experiments that I showed you have really given me a foothold into this problem because they've identified a cellular effector, which seems tied to the evolution of a particular body part. But again, the amazing thing about these species is that we've mapped QTL associated with changes in this trait. So I'm showing these QTL here. This was generated by Eden McQueen, who is a former graduate student, in collaboration with Peter Andolfato's group at Columbia. And in these maps, the probability of association with posterior lobe size is plotted as a function of genomic location. Now, there are a bunch of interesting regions that are spread all over the genome. They're marked with these vertical dashed lines, but dumpy is not in one of them. It's right here in between QTL peaks. And that's actually a really great outcome for me because it suggests that some of these peaks might correspond to dumpy regulators. Now, even though this is a really powerful method, there are still hundreds of genes in these regions. And so to narrow them down in order to map the network controlling dumpy, I've worked with these same collaborators to build a couple more matrices in the form of genomic data sets. So I've done RNA-seq on whole genitalia in both species to find differentially expressed genes. But I've also done allele-specific RNA-seq in hybrid samples, which is what I'm most excited about. And so if you think about a particular gene in a hybrid cell, there are two copies of that gene, one from each species. I can use RNA-seq to detect imbalances in between these alleles, but those are only going to happen if there are differences in the regulatory sequences for this gene, and that would make it a high priority candidate within a QTL. And so with this method, we've found 6 transcription factors that lie within QTL that have these imbalances, along with a bunch of other cellular effectors that may or may not interact with the matrix and shaping the tissue. And I think these resources are going to be really useful for building out the network controlling dumpy specifically, but I'm also going to be able to extend it to other matrix genes that also vary in genital expression. So I'll show you some expression patterns for those. Here are 2 other matrix components that are expressed in the developing lobe at early time points. They're named Neo and Morpheus. I'm not making that up. They're actually named after characters from the Matrix movies because that's what Drosophila gene nomenclature actually allows, although they're deliberately misspelled. This next one, Trinity, is expressed in bristle cells. So there are bristles all over the genitalia, but you can see them most clearly in this bottom structure. And this last one, Cipher, is expressed in the phallus in the middle of the tissue. Now, all of these genes have differences between these species, especially Cipher. Its pattern looks completely different. I have no idea why. And so in the future, I want to figure out how all of these genes are regulated using the method that I described before, and also how they work together in order to shape this tissue. So as I come back to my title image, I want to leave you with a couple of take-home messages. So first, the matrix is everywhere, at least when it comes to fruit fly genital structures. It's made of multiple genes, but the precise role of each gene isn't really well understood. And I'm excited to use this tissue or this system to figure out how they're regulated and how they work together to control multiple body parts at the same time. And second, I've developed all the techniques necessary in order to make Drosophila genitalia computable, just like the embryo. I can resolve individual cells and associate with them with the matrix and with the expression of individual matrix genes. And that's potentially really interesting because in the future, I could use computational modeling to ask more mechanistic questions about what the matrix is actually doing to these tissues and whether that function itself is changing between species. And so just like Neo discovers that he has limitless potential in the Matrix movie once he understands how the matrix is built, I hope I've convinced you that the system really shows a lot of promise in revealing the underlying architecture of gene regulatory networks in development and evolution. So I'd like to thank everyone that helped me with this work, especially my primary mentor, Mark Bays, and I'd be happy to take any questions.
Thank you, Ben, for this amazing talk with a lot of metaphors and citations of our favorite movies, one of my favorite movies. So if you guys have any question from the audience, you can either write in Q&A box or you can raise your hand and I will unmute you so you can ask your question. But there's already a question from Max Stoller in the Q&A. Max Stoller asked, what is the shape of the sword or shield in the hybrid?
Oh, that's a great question. Thanks, Max. So the hybrid lobe is basically looks like the shield in terms of its overall shape. So it's pretty flat, but its length is half the size. So there's some influence from each of them. And in terms of the trait that varies the most, which is basically like how long it is along the surrounding tissue, it's exactly intermediate, which is actually very helpful because it means you could do crosses in both directions. Which can give you some additional candidate genes to figure out what's regulating everything.
So there's also another question from Carmelo Ferrai. I am unmuting Carmelo.
Hello,
Okay.
Hi there.
Hi.
Very interesting talk. I have a question about, you're showing that there is this different gene regulation, but I'm kind of interested more in the physical kind of aspect of this phenomena. Do you know if it's possible, or eventually you're planning to try to check if there is different level of stiffness that are superimposed by the different distribution of the matrix? Yeah,
so I would love to investigate like this and lots of other biophysical properties that are associated with the matrix. I'm not a trained biophysicist myself, and so if anyone has any like suggestions in that area, I'd love to hear them. The closest that we've come is that people in the lab have tried to do basically like cut the matrix using basically laser ablation and detect like small movements in order to get something like elasticity or stiffness. Those, we haven't had like a whole lot of success in terms of like the reproducibility of those techniques. There are also like genetic ways that we can think about doing that with optogenetics, if we could put in a optogenetically responsive element to cleave it at a specific time. So yeah, that's like, I think a real prospect of the system is that it allows a bunch of biophysical measurements and the ability to connect those with the genetics. But I think we have we've got a little ways to go in order to like figure out how to apply those techniques to this system.
But at least, do you know, for example, or can you, ideally what I can see is that you can just try to do some atomic force microscopy in the region that you're analyzing. To show, for example, if there is different level of stiffness on the different edges of the tissue that may be resolved, for example, if you make all your mutants.
Yeah, that's a great idea. I'll definitely look into it. Thanks for the suggestion.
So just to finish that stuff, there was some, because there's some people that, I know that some people does it in UK, for example. They did it even in tissues, like in brain. So it's not impossible to just try to analyze in total, more global level areas of a tissue.
Sure.
Many things is done in cells, but if you use the right cantilever, you can also check for the portion of a tissue.
Sure. Great. Thanks so much for the suggestion. I appreciate it.
Thanks, Carmelo. So there's one more question in the Q&A box. Tanzim Rafik asks, fantastic talk. Have you checked if the pressure imposed by extracellular matrix on sword is different from that imposed on shield?
Yeah, I would love to know that too. So this reminds me of some recent work that's been done in the vertebrate ear that relates kind of matrix components with internal pressure in terms of shaping body parts. Haven't done anything like that yet. I've been mostly focused on basically identifying the effectors themselves and kind of broadly characterizing where they are and seeing if there's any kind of qualitative differences between them. But again, this is like a really interesting mechanical system, and there's a bunch of mechanics that we just don't know yet. And that's another thing that I'd love to think about checking.
I think Pravrusa also had a question. Pravrusa.
Hi, Ben. I really enjoyed that. That was wonderful.
Thank you.
I had two questions. The first sort of follows up on one of the questions that came up before about the hybrids. Can the hybrids then mate with either parent? Are they fertile at all?
Yes, they are completely fertile, which is how we are able to. So basically those QTL maps are created because by doing these back crosses, we basically scramble the genome and then statistically associate markers in the genome to the phenotypes of those progeny. So that's how they're generated. So yes, they are fertile, and we can do back crosses in both directions. I tend to just do them in one direction because it's the direction that produces the most flies. But it should work in both, which in theory isolates the influence of the X chromosome specifically in terms of like how it's shaping the tissue itself.
Cool. And then the second question I had is, it looked like most of the structures you're looking at are in the larvae. I was curious if males and females between these species have different structures, and if the males and females are the structures are like co-evolving for that species.
Yep. So these are all pupal structures. So in the larva, this all comes from the genital disc, which is actually inside out. It everts in order to form this general tissue, and then that tissue is warped later. But in terms of coevolution between males and females, there is a co-evolving female structure, which is called the ovescaped pouch. That does seem to co-evolve, at least among the 4 species in the Melanogaster clade. So the species with the shield has the biggest pouch, the species with the sword has the smallest pouch. And we're really interested in trying to understand if there's any sort of genetic pleiotropy operating here, which could escape or explain that coevolution, or if there are basically like the way that we would think of conflict happening between these structures, which is responsible for the coevolution there. But there's a lot to explore in female tissues. And that's kind of another category of structures that this general system allows in terms of like understanding the mechanics that control their development and their evolution.
Very cool. Thank you.
Thank you.